Hi everyone, again welcome to the structural biology course. Today is the last class of the module MD simulation. In the previous 4 classes, we have discussed about general features, attributes, force fields, conception about MD simulation, what it is, how it is going. In the second and third class, I have discussed in detail of the process. In the fourth class, I discuss about the data analysis. Today, in the last class, I would talk about 2 case studies which will help you to understand that how the knowledge you have acquired could be used in helping your research. The first case study on the application for molecular dynamics simulation is a very short one, and why I put that story, because I wanted to tell you it's not always we perform MD simulation and we publish the MD simulation data. It could even be helpful in designing your experiment. So, the protein you are looking here in the movie is the protein which I have studied in my PhD. This protein is called human deoxycytidine kinase. Human deoxycytidine kinase is very important protein because it is one of the major target in cancer chemotherapeutics. So, people study this enzyme, try to understand, design new drugs and all those things. Especially nucleotide analogues are mostly successful drugs and people design many of them. You name aracitabine, gemcitabine, and mostly the L-nucleoside analogues, they all are used as anti-cancer drug. While studying this protein, I was not someone who used to use or who is an MD simulation expert, neither was my lab. But being in Chicago at the time of 2004, 5, 6, 7, where there was kind of a revolution going on, taking Klaus Sultan in UIUC, Benoit Raux in New Chicago, I also wanted to learn, and with my initial understanding, I have run this protein a 5 nanosecond run, what you are looking at. I know many of you who have now acquired knowledge about MD simulation are probably thinking that 5 nanosecond is not a considerable amount of run, but that time with a 2-core computer it takes huge time. But what I am trying to say, if you look at this carefully, you will understand that this 5 nanosecond run and the movie I have created from this help me understand that, one, the central core of my protein have certain rigidity. So, this part is rigid. There is a big movement of this loop, and this part is also having movement. So, this is minor movement, this is major movement. How these informations were helpful in my research? When I was working, this convinced me that because I see the movements, so there would obviously be other conformation of the protein. So I keep trying with different crystallization condition, and if you see that, we see a clear movement here. We also see a movement here. So we published modified form of this protein by doing crystallography, but the inspiration I got came from my initial MD simulation data. Now I will talk about the lead region. If you again go back, you see the lead region is clearly the mostly moving region. Especially the tip region, if you see, they undergo a huge change. So, the time we didn't have programs to check, I had an idea that there might be a chance of post-translational modification which probably change the conformation. That time we don't have the availability of the programs as server, so I wrote a program and compare the sequence of different deoxycytidine kinases, and I found that at the position 74 there is a serine that have a high chance to be phosphorylated. Getting that idea along with the movement I have seen in my protein structure, I make a phosphomimic. Students who don't know about phosphomimic is, nowadays you see something, there is a probability of phosphorylation, you buy a kit and phosphorylate. That time, being into a crystallography lab at the time of 2005-06, we don't have that expertise or courage. But on the other hand, being structural biologist, we did our own modeling, and if you do modeling, you could find that where you have a serine, if you replace it with glutamate, it kind of mimic the serine in its phosphorylated state. So, we make few mutations to make our logic clear. First, as I told, we make S74 to glutamate. We change S74 to alanine. So, we make glutamate. We make alanine to see what is the null effect. We make S74 to aspartate because we wanted to prove that it is not the charge only, the length, because glutamate and aspartate have same charge but length is less. So, we wanted to compare and we wanted to make glutamine, which will say size, same size but charge. So, what we mean, it would only work if the length and the charge both fit. And when we make a S74E mutant, we get high activity. If you compare in comparison to a S74A, it's really high, but the wild type, when we put it in the cell, it was phosphorylated. So, there is a mixture of phosphorylated and non-phosphorylated, and that is why the difference is So, by making simulation, I have understood and designed my experiment successfully. That is one of the very interesting application. Now coming to another one where I have done a in silico work to make a thermostable carbonic anhydrase designing. So, this is application of molecular dynamic simulation in screening thermostable carbonic anhydrase for industrial usage. First, let me tell you about the project. You all know about the global CO2 emission. If you see this, you will see how it increasing rapidly with years. And coal is the largest contributor to global CO2 emission from energy use, which is 41%, and its share is projected to increase day by day. So, who is unaware about the fact that why coal is used? Let me tell you, 50% of the electricity generated in US is from coal. In 2006, coal-fired power plants produced approximately 36% of the total US CO2 emissions. US produces about 1.5 billion tons per year of CO2 from coal burning power plants. In the US, electricity demand is expected to nearly double over the next 30 years. That means more CO2 emission. Now, why I talk about US? Because they maintain those rules and restrictions the best. So, if this is the statistics of US, for other countries, you could easily understand they are even worse. China and India are rapidly expanding their use of coal for energy generation. Why is coal, the future use of coal is likely to increase? One, coal is cheap. Two, coal is abundant. United States, Russia, China and India have immense coal reserves. The economics and security of supply are significant incentives for the continuing use of coal. So, when CO2 gas is emitting, what are the existing technology? So, existing technologies are inefficient and energy intensive. There are amine absorption, there is calcium hydroxide absorption, and there is gas separation. Among the three processes, amine absorption, calcium hydroxide absorption and gas separation, amine absorption is the best available technology, and for this, the best one, it is economically unfavorable because it takes 50% cost of efficiency. So, you are generating power, 50% of that is needed to absorb the emitting carbon dioxide. So, we have targeted for a project where development of cost effective immobilized enzyme system, we want to develop a biological enzyme which could capture carbon dioxide from power plant flue gas and prevent its emission to the atmosphere. So, what we are trying, if flue gas is coming out, this is having really high temperature. It is not possible for any biological enzyme to be there. If it would be there, it would be denatured. That means it is non-functional. But flue gas, when it is coming out, you could cooling it down, coming it around 90-100 degrees and then go through the screening system of this biological enzyme. So, what we are trying to? We are trying to get an enzyme which could absorb CO2. We want to put it in a support matrix where it would be attached. So, this would be a device like that where there will be support system and there would be enzyme immobilized. This is called enzyme immobilized support device. And our choice was carbonic anhydrase, because carbonic anhydrase is a family of zinc containing metalloenzyme that stimulate the interconversion of carbon dioxide and water into bicarbonate and proton. So, it would convert CO2 to bicarbonate, and further you could convert it to methanol and other products. So, not only you absorb the carbon dioxide, you could develop product out of it. This is the biochemical reaction, carbon dioxide plus H2O through producing bicarbonate. Also, interesting fact is carbonic anhydrase is found ubiquitously in prokaryotic microorganism, bacteria, algae, plants, as well as in higher eukaryotic system. So, this could do, and this is available. These are the 2 points which makes us interested to work on. Before going into the details of the process, if you look at the structure of carbonic anhydrase, it is a alpha beta protein with distinct fold. Mostly if you look at, so there are alpha helices, there are beta sheets. There is a twist. If you see in this portion, you will find that there is a twist. The twist of the beta sheet, and this twist create the active site pocket. If you see, there is zinc, at the bottom of which lies the catalytic zinc. There are several long and flexible loop in its structure which also play important role in enzyme catalysis. So, if you see these green ones are loop. You see that this enzyme is flexible and these loops have very interesting or critical role in catalysis. But at the same time, presence of these long loops would make your enzyme less adaptive to the high temperature. If you look at the active side of the enzyme carbonic anhydrase, you see the, if you look at here is the zinc, this is coordinating with histidine residues, 1 histidine residue, 2 histidine residue and 3 histidine residue, and a catalytic water molecule which is here. The role of zinc in carbonic anhydrase is an electrophilic catalyst. We will take a look on that. So, this is what is happening mechanistically inside the enzyme. The zinc binds to the water and then it deprotonates, so the oxygen become negative and binds to the carbon dioxide. Then there would be a nucleophilic attack by hydroxyl group on the carbon atom of the carbon dioxide, and finally the displacement of the bicarbonate ion by a upcoming H2O. So, that is the reaction mechanism. Our initial action plan was we will clone probable carbonic anhydrase and then we will screen them based on specific activity and thermostability. Then when we get a good one, we could use biochemical engineering approach to scale it up and make the enzyme in high amount and then immobilize it in the device and use that as a screener or absorber on the exit side from where the flue gas is coming out. So, clearly there are 2 phases, one is protein biochemistry, second one is biochemical engineering approach. So, we want to screen carbonic anhydrase with high enzyme activity and high thermostability. But the whole process was high cost demanding and practically impossible, because you have to bring new enzymes, you have to clone each and every one and then test their specific activity, thermostability. It demands a lot of money, lot of manpower, and kind of says that it is impossible. So we decided to replace the whole thing with molecular dynamic simulation. What we wanted to do? What we wanted to do is, we are looking for such carbonic anhydrase enzyme which is stable both thermally and pH wise, so that it could be used industrially to reduce excessive carbon dioxide emission. To achieve that goal, we are developing in silico screening instead of in vitro screening, which we planned initially. We alter it to the in silico screening process and approach of understanding the structure stability regarding those stability criteria. In the study, to understand the stability and dynamism of carbonic anhydrase, we decided to perform molecular dynamic simulation at different temperature gradient. So, let me explain the plan in more elaborated way. We already know that one alpha carbonic anhydrase found called sulfuri hydrogenobium species is having very high stability. We know carbonic anhydrase from Neisseria gonorrhoeae with very high activity. So, as I told, high activity Neisseria gonorrhoeae alpha carbonic anhydrase, which we call Ng alpha Ca, and thermostability sulfuri hydrogenobium species, which we call SSP alpha Ca. We decided we will understand the reasons of thermostability from SSP alpha carbonic anhydrase, and those understanding we will use to make Neisseria gonorrhoeae alpha carbonic anhydrase engineered towards thermostability. This is a comparison of sequence and secondary structures, where we have taken informations of active site. You will see the green boxes, interacting triad, which is the red boxes, the hydrophilic residues, which is the sky blue, the hydrophobic ones, which is purple, and disulfide bond forming residues, which is orange. So, you get a idea about the protein. These are two proteins, and if I align one protein into another, you will agree with me that the alignment shows really high structural similarity. So, these two proteins are really structurally similar, which means if we get the knowledge from the SSP alpha Ca, it could be easy to apply to Ng alpha Ca. Also, when we compare their secondary structures, we see very similar profile between the two. So, we go for MD simulation techniques. We perform MD simulation at 4 different temperatures: 300 Kelvin, 350 Kelvin, 400 Kelvin and 500 Kelvin, and have done for 100 nanosecond time scale. MD simulation has been performed in GROMACS 4.5.4 version using AMBER 99 SBILDN force field. Generally, these we use for only protein. We use TIP3P water model, and the energy minimization was performed using steepest descent algorithm for 50,000 steps. And as we told, all the runs were run for 100 nanosecond in term of production run. So, this is the analysis of the result. You see that when we compare the root mean square deviation, we see that from 300. In 300, interestingly, we see more flexibility on the thermostable protein, whereas when it shifted from 300 to 350 and further, we see that gradually there is more RMSD values in case of mesophilic protein, which was expected. We have also checked the principal component analysis PCA, and if you see, we have performed 300 Kelvin. In 300 Kelvin, again you see the mesophilic protein, which is the Ng, is smaller than the thermophilic one, but when it changed from 300 to 350, the PC 1 and 2 graph, the projection or eigenvector 1 and 2 had increased significantly, whereas relatively less increase we have seen in the thermophilic one. When we come to 500, you will see it opens up, and it opens up radically at coming at 500 Kelvin. We have also taken a look to the C alpha motion onto the first and second eigenvector, and here also we see similar trend. We look at the free energy landscape of Ng alpha Ca. As I told, these are the free energy landscape representation, where PC1, PC2 are in 2 dimension and free energy is in 3rd dimension. We see that the graph is expanded, and at the initial stage the lower ones were extremely sharp, that become broadened in higher temperature. Similar trend observed in the thermostable protein, but in a much reduced scale in comparison to the mesophilic protein. When we take a look at the comparative RMSF, we see that the flexibilities are really high in case of Ng alpha Ca in comparison to SSP alpha Ca. In the loop region, the threonine 5 to serine 14, serine 22 to phenylalanine 25, glycine 31 to isoleucine 50, valine 95 to proline 107, leucine 163 to lysine 168, glycine 174 to valine 185, glycine 209 to glutamate 226 are very flexible in Ng alpha Ca, which is the loop region. We have done individual residue analysis in terms of their fluctuation values. We have also checked the salt bridges. Salt bridges are non-covalent interaction, but because of the development of real charge, the interactions are really high energy when you compare between the non-covalent interactions. So, in case of Ng alpha Ca, we see that the interactions happen mostly with 2 amino acid, the pair arginine 136 and aspartate 194. So, we have observed one salt bridge formation development in case of Ng alpha Ca, whereas in case of SSP alpha Ca, we see formation of salt bridge between lysine 36 and glutamate 99, lysine 41 and glutamate 223, lysine 39 and glutamate 223, lysine 136 and glutamate 133, arginine 165 and glutamate 223. More importantly, if you see that they are not restricted in one place, they are actually spreaded all over the structure. This is the representing movie we have found by getting the MD simulation trajectory. If you take a careful look on the movies, you will see that in case of mesophilic protein, this is taken at 350 Kelvin, you will get more pronounced effect at 400 and 500, but here also you could clearly see that the mesophilic protein expanded, whereas the thermophilic protein is more into its maintaining its structure. That clearly shows the difference of structural integrity between the thermophilic and mesophilic protein. So, simulation help us to understand the difference of movement in this mesophilic and thermophilic carbonic anhydrases. Important non-covalent interactions like salt bridges are showing their effect in a time dependent manner. Identification of flexible residues helps in designing thermostable protein. So, with all those we start designing, and the first idea was to incorporate some salt bridges. So, one strategy was to take the serine 44 residue, change it to arginine. Lysine 168, this is very important. If you see, lysine and arginine both are positively charged, but looking at the interaction, it seems that if we change lysine 168 to arginine, because of its double spearhead interaction, much more stable interaction is possible. And also we change serine 139 to glutamate 139. So, before going into the simulation story, I just want to show you the effect of mutation on the position serine 44. The serine 44 individually have no interaction in the mesophilic protein. In the thermophilic protein, the equivalent position is lysine 41, which shows interaction with glutamate 223. Whereas the mutant version of the mesophilic NGCA, where we mutated the serine 44 to arginine 44, we have observed 3 interactions of arginine 44 with the glutamate 226. Based on our analysis, we have also determined the melting temperature. The melting temperature shows that for mesophilic protein, the melting temperature is 312 Kelvin, whereas this is shifted to 348. This is 348 Kelvin, 36 Kelvin higher in comparison to the mesophilic protein. Only introduction of 3 mutation changed this melting temperature from 312 to 333, an increase of 21 Kelvin. Now, when we are comparing the MD simulation data of the mutant protein, we see that it shows enhanced movement in comparison to the same protein in its non-mutated form. Also, this is the movie comparing the mesophilic protein and the same mesophilic protein with the mutant. Again, taking a careful look, you will understand that all those loose motions which were present in the mesophilic protein now absent because of the development of those 3 mutations. So, in summary, what we did, we use MD simulation to compare the dynamic properties between mesophilic Ng alpha Ca with thermophilic SSP alpha Ca. What we looked at? We looked at mostly non-covalent interactions, looked at flexible residues, and based on our understanding we make 3 mutations. These 3 mutations change the stability of the Ng alpha Ca. And if you look at MD simulation method, gives you opportunity to give a look on your protein which is not possible with any other techniques. So that is one of the beauty. Here you could look at the dynamic property, you get into another angles to analyze your protein, you could look at the intrinsic flexibility in your protein, and all this knowledge, especially if you have ligands, which I am not showing here in this case study, if you have ligand, how it is bound, how it is stabilizing, how you could alter, all this would be very critical towards enzyme engineering, towards drug designing and whatnot. So we are on the edge of finishing this module. I agree this is the introductory course, so the things we have discussed would not make you an expert in MD simulation, but I have tried my best to talk about the basics of the process, the components. I start with a very basic question: why and what is MD, what is simulation, how simulation mimic experiment, what type of experimental system it mimic, and how it proceed further, how the time scales are there, what type of physics is used, how the force field are, how you could influence a protein by developing a force field which is competent to that, what are the components, how they could be used through an algorithm like Bartlett, velocity Bartlett, lift fog and all those algorithms, by allowing the Newton's law of physics, and then how from a sequence you would start your journey to get a structure, repair those structures, modify those structures, come to the process of simulation, then you again make things like you add hydrogens, you add a box, you add ions, and you make a system experimental like mimicking of an experiment. Then you go for minimization, energy minimization to make the bad contacts better, and then go for equilibration with the ensemble conditions, and finally to the MD simulation run. I have also discussed several analysis methods which will be helpful for you to perform, and then I have discussed case studies. I will finish up just by mentioning that in this introductory course, in this limited opportunity, what I have discussed, we call it classic molecular dynamics. But the field of MD simulation in protein is not restricted in classic molecular dynamics. Let's discuss about that. You remember this, I talked about we have initial coordinate which is prepared for simulation, then we add hydrogen, then we add a new box, then we solve it, add water, then we add ions, and we got final structure. From there we should possibly have bad contact, so we have to do minimization. So we start with the J ion and we go for minimization, energy minimization, which helps finding the nearby local minimum. But then from the local minima, equilibration helps us escaping the local energy barriers, which is small barrier. Then the classic MD run helps us to come to the global ones and it sample thermally accessible states. Now from there it potentially looks like this is the end of scope of simulation, but it is not true. Whatever we do in MD simulation, we compare between the thermal states, but we could go beyond that by applying the steering forces which are needed to access other intermediate states in a timely manner. So if you want to study conformations in different states, if you see these are energy barriers, we could have to do or perform something which called bias or targeted molecular dynamics. Nowadays there are many popular methods are coming. As I told, in the scope of this course I cannot go beyond, but I will talk about them so that interested people could go and learn them. Steered molecular dynamics, which is very critical, especially to understand the interaction between macromolecule and small molecule. Replica exchange method by resampling samples from different temperature. Metadynamics, multi-scale simulation, as we think that if we start from one point, it is difficult to go to the next conformer. So what we do, we run a simulation or we get different conformers and apply simulations to all of them. Is multi-scale simulation. Quantum mechanical, molecular mechanical hybrid method, and coarse grain if you do not need on the atomic level. If you want to look at the movements of bigger structures, a whole loop, a whole structure, then coarse grain is your method. I would finish by talking a little bit about one of the favorite method of mine, which is hybrid QM-MM approach. As you already understood, by applying Newtonian physics, you cannot perform bond breaking and making, but when you are studying an enzyme, it is very critical to look at its conformation while the chemical reaction is happening. But when you have to study the breaking and making of bonds, you have to make quantum mechanical calculations. Is it possible to perform quantum mechanical calculation of the entire protein? No. Because if you think, even if you consider a small protein, 200 amino acids, it would have the number of electrons in such a high number with their cross interaction that even the best supercomputer the world is having would take innumerable amount of time. To solve this problem, scientists have come up with a hybrid method which is called hybrid quantum mechanical molecular mechanical approach. The development of this approach is guided by the general idea that large chemical systems may be partitioned into an electronically important region which requires a quantum chemical treatment and a remainder which only acts in a perturbative fashion and thus admits a classical description. So, if you look at here, this is the whole system is treated with the molecular mechanical approach and the small portion is treated with a quantum mechanical approach. Let us say now you are talking about an enzyme, the amino acid side chains of the active site and the substrate would be in the quantum mechanical region. Rest of the protein which shows movement is in the molecular mechanical region. So, if you calculate the energy now have three components: energy of the quantum mechanical region, energy of the molecular mechanical region and energy of the quantum mechanical molecular mechanical region. The energy of the quantum mechanical region would be calculated using Hamiltonian operator. You already know how to calculate the energy of the molecular mechanical subsystem. The top part is how to calculate the QMMM portion, because you have to consider the interaction between the QM and MM region. There are several approaches considering a link atom, considering ghost orbital, and all of them I am again not going into, but I am just showing you the possibility and the successful implementation of a calculation of a biochemical reaction. So, there would be three layers: one small model system, the first layer, bond formation, breaking take place, need to use high level method, the quantum calculation. Intermediate model system, second layer, electronic effect on the first layer, need to use medium level method, and real system, the third layer, environmental effect on the first layer, need to use low level method. So, like this we have to go in details about all the processes, and knowing about them, understanding them would help us with more options to develop more realistic model and their calculation of biological macromolecules. Thank you very much. I am finishing this module here. Thanks for listening. Thank you.